From tiny bits of matter, nature assembles the amazing structures of the physical world. The air we breathe, the water that sustains us. Even living organisms are all assembled from simple atoms and molecules. Scientists find little difference between the atoms of a tree and the soil from which it grows. So how is it that some atoms form life, while others do not? The answer lies in the structure of these chemicals and how they interact. Nature composes an unimaginable variety of life forms from simple chemicals. Over time, these atoms and molecules flow from one form to another in an endless cycle that sustains all living things. But while some chemicals support life, others can destroy it. This mountain wilderness may seem untouched, but inside these trees, a chemical war is raging.
This sapling's having a tough time. You can see by the yellowing on the needles. That's a diagnostic injury for ozone damage, air pollution damage on this tree.
Biologist Mark Poth is studying how air pollution attacks these mountain pines so he can develop better ways to defend them.
These trees have received toxic levels to the plants of air pollutants since sometime around World War II. What they've received is a full assault of everything that we put in the air. All of this has had a cumulative effect on the forest and has caused some problems.
The damage begins at a level too small to see, with the atoms and molecules that compose all matter, and therefore all living things.
Matter is really anything that takes up space and has a mass. Virtually everything we see around us is matter. Even things we don't see around us are matter. The air that we breathe and all the components of it.
Imagine we could see the smallest units of matter, atoms. Scientists identify atoms by the number of particles they contain. The core, or nucleus of an atom, contains the heavy particles, the positively charged protons, usually joined by neutrons which have no charge. Circling the nucleus, we find the exceptionally lightweight electrons, which carry a negative charge. These basic particles make up the 92 different atoms found in nature.
The elements are defined by their building blocks, starting from the smallest of the elements physically and by mass. That's hydrogen, and it consists of one proton with a positive charge and one electron with a negative charge. As you move up in size, you add more protons, more electrons to balance the charge for any particular element.
In a neutral atom, the number of protons equals the number of electrons. Hydrogen atoms have one proton and one electron. Nitrogen atoms have 7 protons and 7 electrons. The number of neutrons, however, can vary, creating what scientists call isotopes. For instance, a single neutron makes the difference between hydrogen 1 and hydrogen 2, or nitrogen 14 and nitrogen 15. Isotopes are atoms of the same element, but with different numbers of neutrons, and thus a different mass or weight.
The isotopes of any particular element really don't react differently, and that's where we can use them to our advantage. Since we have techniques that allow us to measure the isotope, we can find out where a particular element is going, particularly if it's important for tree nutrition or a pollution problem, perhaps.
Because pollutants carry more heavy isotopes than chemicals found in nature, Dr. Poth can measure exactly how much pollution the trees are absorbing.
So we can take a look at isotope compositions of nitrate in stream water, look at the isotope composition of the oxygen and the nitrogen in the nitrate, and make some judgment about, well, where is this coming from? For our experiments to look at the effects of air pollutants on plants, we use controlled exposure chambers like this. This experiment is set up to look at nitrogen compounds, in particular nitric oxide. There are a couple of different ways to measure the effects of the pollutant. One is to directly measure effects on growth, either the stem diameter, or we can take the calipers and actually measure changes in the length of the needles very accurately.
All plants require nitrogen and oxygen to survive, but these two elements can combine to form a dangerous compound called nitric oxide.
Compounds are made up of different elements combined chemically so that they form something completely different than their pure elements. The types of compounds that we see in air pollution can be complex mixtures of many different elements. For instance, ozone is made from 3 oxygen molecules, and it's a very reactive air pollutant and very aggressive. It's what makes our eyes sting, our lungs hurt, and damages trees directly.
How atoms combine into compounds is determined by their outermost particles. Electrons fly around the nucleus of an atom in specific regions called energy levels or shells. Each energy level can hold a maximum number of electrons, 2 in the first shell, then 8 in the second, and so on. Each atom has a tendency to keep its outer energy level full, but some atoms have to steal electrons to do it. For instance, a sodium atom has just 1 electron in its outer shell. It easily loses this lonely particle to a nearby chlorine atom, which is missing just 1 electron in its outer shell. The transfer of this negatively charged particle leaves both atoms with complete outer shells. But it gives the sodium a net positive charge and the chloride a net negative charge. These oppositely charged atoms, called ions, attract each other, forming what scientists call an ionic bond.
The first rainfall of the season, the soil is very dry, so it takes the ions from the ionically bonded solids into solution, brings them into solution, moves them to roots, makes it available for uptake and use by all the plants. The ions that are within the soil that are important for the tree's nutrition or the plant's nutrition and making up life. And that's going to include things like calcium, which is used structurally to stabilize the structure of the tree. It's also used physiologically in the cell. Potassium, magnesium, phosphorus, all of these are going to be important. Nitrogen and sulfur for making proteins, the machinery of the cell.
Instead of stealing electrons to form ionic bonds, many atoms share electrons to form what scientists call covalent bonds. In a molecule of hydrogen gas, 2 hydrogen atoms share their electrons equally. The same is true of the atoms in oxygen gas, because neither of the pair is stronger. If oxygen binds to hydrogen, however, the electrons spend more time near the larger atom. This makes the oxygen partially negative and the hydrogen partially positive. Unequal sharing makes this covalent molecule polar, which can lead to some interesting attractions.
Living organisms take in the elements, ions, and molecules of nature and assemble them into their own more complex carbon compounds called organic molecules.
We can see a dramatic example of the presence of organic and inorganic materials in forest soils. If you go to an area where there's been a recent road cut and take a look at the surface layers where all the plant material, or the majority of the plant material, is deposited. The leaves fall, the fine roots grow through these surface layers and then decompose later. You can see a darker tone to the soil there, and that's the organic material that's been incorporated into the surface of the soil. The deeper upper layers are coming from the rock parent material that the soil's forming on, basically inorganic constituents formed geologically. And so you get an interesting contrast presented to you between organic and inorganic compounds.
Airborne pollutants don't stay in the air. Rain washes these chemicals from the sky and into the forest soil, where they do further damage to the trees.
Here's the soil samples I wanted to show
By
you.
carefully measuring both sources of pollution, Dr. Poth hopes to find ways to protect the survivors of this chemical warfare.
Many of the young trees are going to have a chance to survive in that many of the trees that we see are from those resistant trees that made it through that initial air pollution episode that was so high before we had really emissions controls on cars and many of the other things. They do represent sort of the children of the survivors.
While some chemicals threaten life, others form the basis of it. As atoms combine, they produce molecules that support all living things, including one compound so crucial we could not survive without it. A single compound dominates the living world: water. With it, life flourishes. Without it, life struggles to survive. Water is the major constituent of all living organisms. It covers almost 3/4 of our planet, yet most of it is too salty for humans to drink. So many have turned to the promise of desalination to make the ocean a freshwater resource.
This process really is short-circuiting Mother Nature. Right now, as we look at the hydrologic cycle, water is evaporated out of our oceans, it forms into clouds, it travels over land masses, and then it falls as rain or snow on the land. It falls and works its way across the land into rivers. The rivers go back into the ocean. But in the meantime, we tap into those rivers and we use that water. And this has been done since the beginning of time on Earth. The water we're drinking today may well be the same water a dinosaur drank millions of years ago. Instead of waiting for the sun to heat the water, we're heating it ourselves. Instead of waiting for the clouds to move over the land, we're condensing that water ourselves, and we're using it right at the point that we're taking it from.
The desalination of seawater is not a new idea, but the high cost of heating water makes it impractical for large-scale use. The reason lies in the chemical nature of water itself.
Water is basically a compound. In water, we have both oxygen and hydrogen, both of which are elements. When they join together, they form a compound. You can find oxygen on the periodic table, you can find hydrogen on it, but you can't find water on it. The bonds in water are polar because the electrons from the hydrogen are drawn very close to the oxygen, giving it a slight excessive negative charge and leaving the hydrogen with a slight excessive positive charge.
This polarity means the hydrogen atoms of each water molecule attract the oxygen atoms of others, an example of what is called hydrogen bonding. These bonds create the surface tension that holds together a drop of water and allows an insect to dance across a pond. Hydrogen bonds also explain why water requires so much energy to boil into the steam necessary for desalination. But Snyder's team has minimized that problem by using another property of water to their advantage.
It will actually boil at a lower temperature as the pressure decreases. As an example, if we were to take water and put it under a full vacuum, that water would boil at under 40 degrees Celsius. And that allows us to recover the maximum amount of fresh water for the amount of energy we have to put into that water to heat it.
As the fresh water evaporates away, the water and salt left behind become a hot brine of highly corrosive ions.
Ions have a unique property in that they can actually penetrate the surfaces of many metals. When we go into a desalination plant, this property of salt water that can cause the rusting and the corrosion of metals is a very high concern, especially when you're dealing with salt water, seawater that is at the 100 degrees Celsius, very hot seawater. It becomes even more corrosive as the activity within the molecules increases. So we're dealing with that by using some very high-tech metal alloys, aluminum alloys in this case. We bring that seawater into the plant. We treat it with a couple chemicals. We also treat it with preventative of any bacteria growth within the system. It then goes through an ion trap. The ion trap is nothing more than a bunch of scrap aluminum that's in there to attract those stray ions and hold them back so they won't hurt the aluminum that's actually in the treatment plant itself. After the water passes through this ion trap, it goes into the real heart of the system. This is the evaporator. This is where the heat is applied to the seawater, where it's allowed to evaporate, and where that vapor is condensed and condensed into fresh water. And really, this is not much different than the good old-fashioned whiskey still that may have been used during Prohibition to make your own backyard whiskey. The process is pretty much the same. The water is heated, it's condensed, and is used. The goal that we have right now is to try to reduce the cost of desalinating seawater by about half of what it is worldwide. If we can do that, we have a tremendous potential here to use that resource.
Mr. Snyder hopes to cut costs by locating his facilities near power plants and using their waste heat to help vaporize the seawater.
It's kind of a symbiosis between the two processes. The real economy comes when we can take this new technology we're developing, couple it with a new power plant or a repowered power plant or an existing power plant, and use those two in concert with one another. Each one of these sites could be a candidate for a plant of up to, say, 75 million gallons of fresh water a day could be produced here. And when you look at that, that's fresh distilled water, pure H2O. So the benefits worldwide of this technology that we're currently developing can be extreme.
Water brings life. Without it, we could not survive. Yet water is only one molecule upon which life depends. By piecing together other compounds, living organisms create larger, more complex molecules, the intricate structures which bring ordinary matter to life.
Okay, guys, got everything?
Yeah.
Tools? Water?
Sure.
Let's do it.
All organisms are chemical beings. Every move, each bead of sweat, even our thoughts are the result of chemical reactions. Yet scientists can explain only a fraction of the many reactions we depend on to survive.
I think a lot of the quenching of the fluorescence is due
Dr.
to that
Susan Taylor
tryptophan.
is a molecular biologist, a kind of biochemical detective, searching for the clues to unravel these molecular mysteries.
Most types of science, you obtain clues as you go along, and you get little pieces of information gradually, whereas crystallography is a long process, and either you end up with a structure at the end or you don't.
Using a process called X-ray crystallography, Dr. Taylor and her colleagues reveal the structure of compounds. With these atomic fingerprints, she can deduce how organic molecules interact. Her work focuses on one of 4 groups of compounds so crucial to survival. Biologists call them the molecules of life.
It's pretty good, yeah.
Carbohydrates, lipids, nucleic acids, and proteins.
Carbohydrates are composed of repeating units of simple sugars, such as glucose. These units can be broken down quickly to provide the fuel our bodies use to function. Lipids are greasy compounds, including those we call fat. With their 3 chains of fatty acids, these molecules offer the body a rich store of energy, more than twice that of carbohydrates. Nucleic acids are chains of repeating molecules called nucleotides, which contain phosphate, sulfur, and nitrogen. In their unique sequences, nature stores the genetic information of our DNA. Proteins are composed of organic molecules called amino acids. Amino acids contain an amine group and an acid group, which are the same in each molecule, but the third, or R group, can differ, making over 20 different amino acids in all. As structural compounds that build tissue, or enzymes that assist metabolism. Proteins perform the work of the cell.
Myself, I'm a protein chemist. I purify proteins. I look at their functional sites. Functionally, proteins are the kind of workhorse of the cell. They digest our food, they convert it into energy, they then use that energy to do other functions, whether it's thinking or motion or storing energy. Everything is done by the enzymes and proteins in the cell. The shape is important because most proteins are catalysts, so every protein contains a string of amino acids, and that unique configuration of the amino acids causes that protein to fold up in a very specific way.
Living organisms assemble small molecules like amino acids into large molecules like proteins through the process of condensation. The different amino acids attach one link at a time, building what scientists call a polypeptide chain. The specific sequence of amino acids determines the identity and function of each protein. Most chains fold and bend into a three-dimensional shape, assisted by the attraction of hydrogen bonds. Some amino acids avoid water and move toward the center, while others prefer water and position themselves near the surface. This molds the protein into a structure tailor-made for a particular chemical reaction.
These are big molecules, and there are many regions of the surface that are important in the docking, and each part must be correct. It's like the key that fits into the lock. And if even one of 100 amino acids is changed, it won't work right. And it won't either bind right, or it won't cause the inside part to be activated. The key must be exactly right, the lock must be exactly right, and if either one is defective in any way, then you don't get the biological response.
For almost a decade, Dr. Taylor worked to map the structure of just one in a class of proteins called kinases.
Well, protein kinases in particular are important for regulation. They turn on and off the enzymes that are in that cell and regulate which ones are functional at any given time. They're critical switches. They must be turned on and off properly. Protein kinases are turned on in response to adrenaline. So when you have a fight or flight response, you need to move quickly, you want quick energy. The adrenaline is secreted into the bloodstream, and it circulates, and then it binds to specific cells, and it activates the protein kinase, say in the muscle cell, to break down the glycogen, the energy that's stored there, to be used. And there are many diseases that are associated with defects in protein kinases. Some types of diabetes are associated with defects in the protein kinase receptor. There are many cancers that are caused by a particular protein kinase that has been genetically altered so that it can't be turned off. That cell just continues to grow. It doesn't stop the growth process.
To reveal how these proteins malfunction, Dr. Taylor launched her investigation into their structure. But first, she would need to synthesize crystals of pure kinase.
Initially, you need to make large amounts of protein, which is something that I can do well in my laboratory. So it's a lot of trial and error, trying different conditions, different complexes, until you find a crystal that is a suitable one. We're going to hit the crystal with X-rays and look at the diffraction pattern. And each of those took us several years to do.
By shining X-rays at crystals of the purified protein, Dr. Taylor hoped to reveal the faint shadow of the molecule's structure. This process once required months of tedious lab work. Then a colleague of Dr. Taylor's, Dr. Nguyen Hue Song, developed a major breakthrough in X-ray crystallography.
Previously, one would have to spend 6 months or more collecting data on many crystals, and because of the technology that Zhang's laboratory has developed, one can now collect data from one crystal in a couple of hours, and that will be sufficient to allow you to solve the structures.
Using Song's advanced technology, Dr. Taylor became the first to solve the structural mystery of protein kinase, which may, in turn, uncover this molecule's important role in the process of life.
Ultimately, the three-dimensional structure allows us to understand how these molecules function. For instance, with the protein kinases, they are switches. They must be turned on and they must be turned off. I think in understanding how they function normally, you can begin to understand how they function when they're defective. So by having a three-dimensional structure of this molecule, it's a first step to rationally designing an inhibitor that you could then artificially turn off a kinase, which can no longer turn itself off. And it's certainly the essential first step in that direction.
Well, the insulin receptor, there's a large insertion.
By revealing the structure of this protein, Dr. Taylor has given scientists a better understanding of life's processes at the chemical level. Atoms form compounds, which in turn assemble into the larger molecules of life. In these complex interactions, we see the processes that build and sustain the spectacular diversity of the living world.